TopMedTalk.
Well, hello and welcome to TopMedTalk. We're coming to you live from Dunleary Harbour, which is in Dublin in Ireland. We're looking out at the window now at cargo ships and passenger ferries heading to foreign shores. And we've been kindly hosted by the British Journal of Anaesthesia, who are having their annual general meeting here in Ireland, Andy.
Yeah, it's great to be here, isn't it, Kate? We've had a really busy day, quite insightful strategy meetings, discussions about journal publishing, processing, huge amounts of things to talk about, but all in a really nice, supportive, constructive atmosphere with pretty nice views out the window of the sea.
Always good. And we were discussing today translational research, how young investigators get into this area, how they progress their career, where they publish their work, who will fund it, and really what the future of it is in the current research environment.
And there's a number of challenges in that environment, but I think also a number of opportunities. And we've been discussing those at length today. And that leads on very nicely to our guest today, who's got a lot of experience in the publishing world, but also in what hopefully is going to be a growing area for the British Journal of Anaesthesia and other anaesthesia publications of translational research.
Great. Well, thank you for the introduction.
So can you tell us a bit about yourself, Gareth Ackland?
So I am based in Queen Mary University, London. I'm a professor of perioperative medicine, and I guess I wear three hats. My clinical work is in anaesthesia and surgical critical care at the Royal London Hospital. And my research is both translational and quite basic science. The basic science is funded by the British Heart Foundation, and the main area of that is looking at autonomic physiology and specifically the autonomic regulation of exercise capacity. And that is undertaken in both laboratory models and human translation models. And the third arm of my existence is translational medicine in the perioperative arena. And that specifically really focuses on organ dysfunction after major surgery. And I'm sure we'll come to the reasons why that's important, but it very much lends itself to exploring human physiology in very granular and highly controlled circumstances, which I think is pretty unique to the specialty of anaesthesia and perioperative medicine.
So for all of our listeners, could you provide just a basic update on the role of the autonomic nervous system in exercise? And then we'll have a great foundation to launch from.
Yeah, so I think everyone will be familiar with the idea that people who are fitter have lower heart rates. And for probably the best part of 150 years, that has been attributed to the fact that the heart rate or the autonomic nervous system of the heart is remodeled during exercise because you're fitter. But the program of work that I co-lead with Alex Guerin in University College London basically just asks the question whether that physiology or the neuroscience of that physiology is upside down, and actually whether it is autonomic control driving exercise capacity, not the other way around. So, and the reason that's important is because one, heart rate is a consistent predictor of all-cause mortality across pretty much every sphere of medicine and actually biology. And so understanding why that consistent relationship occurs is pretty fundamental for unlocking a lot of mysteries of why exercise capacity seems to map to better healthcare and general physiology, ranging from prevention of cancer to having a better outcome from surgery. So essentially that story started when, really with the advent of modern neuroscience, which now allows you to, with unprecedented specificity, to be able to switch off and on neurons at will in a living animal, and actually it's achievable in humans as well. And so basically the work embarked on trying to untangle that conundrum of why lower heart rate maps to more fitness. Anyway, to cut a long story short, in using modern neuroscience techniques, because heart rate is primarily determined by, resting heart rate is primarily determined by vagal outflow, the kind of obvious experiment to do was to stop vagal outflow in a reversible way or augment it and see whether that had an impact on exercise capacity. And so using optogenetic and chemogenetic techniques, you can switch off specific neuronal populations in a laboratory.
Sounds what those mean, Gareth, for us, just for some of our listeners who may not be familiar with those terms.
Sure. So basically, in your brainstem, there are organizations of neurons that control autonomic function. And in a specific part of the brain stem, dorsal vagal motor nucleus, the neurons are essentially genetically coded to be highly specific if you introduce a virus, which then you can pretty much encode genetically for those neurons to produce different receptors or kill them in different ways or reprogram them. And so what that work embarked on was to transfect those neurons with a specific virus with a promoter, which basically meant that could only be expressed in those neurons. And then those neurons expressed a receptor, which you can switch off by giving a pharmacological ligand. Conversely, if you want to activate those neurons, you can transfect them with a virus that expresses a light receptor, so channel rhodopsin, which if you then shine light on it, you basically mimic neuronal activity remotely. And so basically what those experiments were able to do, or that technique allows you to do, is switch off specific populations of neurons at will and test to see what the effect of that is on whatever phenotype you're interested in. And so what that work showed was that if you switch off this population of neurons, you reduce exercise capacity by 80% in a fit, healthy laboratory model, and that's reversible. And alternatively, if you activate those neurons, so essentially what the experiments did was mimic exercise on a treadmill by activating the neurons. And the effect of activating those neurons was the same as training a rat in a treadmill. So in other words, the first evidence that autonomic regulation of exercise was pivotal in determining exercise capacity. That was a key finding, and it explains a lot of the association between these autonomic or heart rate patterns and fitness. And also the consequences of that are quite profound, because it might very well explain why exercise is actually beneficial, because it's actually a neural mechanism underpinning it, not some other factors peripherally, for example. And so we've gone on to explore that now in other translational models, including humans, by trying to mimic that optogenetic and chemogenetic stimulation. And we've done this by work funded by the British Heart Foundation. In fact, all of the work was funded by the British Heart Foundation. And what that has attempted to do is try and activate autonomic pathways in humans, healthy volunteers so far, and sort of hijack the autonomic nervous system to trick it into changing its activity. And so what we did was we asked volunteers to see how fit they were by being on an exercise bike, a cardiopulmonary exercise test, which I know you've talked about in this series before, and then randomize them to either a sham or active stimulation of a specific afferent pathway that actually exists just inside the ear, called the tragus area of the ear. And basically what we found was that in a week, active stimulation increased a measure of your peak fitness, so peak oxygen consumption by 1 ml per kilo per minute, which is kind of impressive for a non-invasive intervention that is, for a short period of time. So it's basically the proof of concept that modulating autonomic function may have a direct impact on exercise capacity. And the work we've just started now is essentially looking at how autonomic control affects exocrines and trying to piece together the impact of that on molecules that we know are released because of exercise, but actually the precise mechanisms that underpin their release is not clear. And the reason why that's important is because it could very well be that many drugs that have an autonomic effect in cardiology and cardiovascular medicine actually have off-target effects on exokines that can be exploited.
So as you've been explaining that, which is just fascinating and not something that I'd considered before, I was thinking that everything I've learned about exercise has been about oxygen and oxygen flux and mitochondria and training muscles. And is that all just irrelevant, or are there multiple intersecting mechanisms of exercise capacity?
Yeah, so I think the central control is the brain. So actually there are some classic experiments done in the 1970s. So you'll probably be aware of these data that, people anticipate their exercise, the onset of exercise. You breathe harder before you've even done anything. That is purely a cortical autonomic response. We were really fascinated by how you explain the central component of exercise performance. And we repeated these experiments in lots of different ways in laboratory models. You basically get the same answer. So we think it is a fundamental feature of exercise capacity, but you're right. Could that be an interaction between that central autonomic control and peripheral physiology? So actually we just published a paper in Circulation Research this year that goes some way to answering that question. The working hypothesis for that was that cholinergic vagal outflow was key to conferring these benefits for increasing exercise capacity. So in a series of experiments, we took genetically modified mice and essentially removed the genes or knocked down the genes that encode for the M2 muscarinic receptor and the M3 muscarinic receptor, and basically dissected the cardiac component of that and the peripheral component. And to cut a long story short, genetically modifying muscarinic innervation of the heart had no impact on exercise capacity. But when we removed endothelial M3 receptors through essentially quite complex genetic manipulation in mice, we found that you get a very similar pattern to what I just described with the zapping of the neurons. And that was essentially when the M3 receptor is absent in the endothelium, the lining of the main blood vessels and actually muscle, you dramatically reduced exercise capacity. The kind of working hypothesis is that there's a direct peripheral link with possibly overspill or some other hemodynamic change that enables muscle to pump or have more blood flow. An essential component to the whole exercise capacity story has always been the muscle pump theory, which is basically you increase blood pressure and heart rate because you have increased flow back to the heart. And actually in the original experiments I described, that was dramatically reduced. We had telemetered animals where you could measure blood pressure in conscious animals while they're exercising, and that was clearly obtunded. So if the M3 muscarinic receptor is absent in endothelium, exercise capacity is dramatically reduced by about 60%, 70%. And so a very likely explanation of that is that obviously muscles have to dilate when you exercise. And the key mechanism of that dilatation is actually mediated by the M3 muscarinic receptor. That's been known for a while, but what wasn't really appreciated was what its functional significance was. So at rest or non-exercise, in a mouse that doesn't have that receptor, they're physiologically indistinguishable from their control littermate, the ones that don't. But when you exercise them, it becomes very relevant, which would make a lot of sense because the only time you really need to have that dilatation is during an exercise challenge.
And the data you mentioned that shows about this central effect happening in anticipation of the exercise starting.
Yeah.
And again, it's a while since I looked at that data, but I'm aware of it. But my understanding is that's a trainable phenomenon. So the animals, whether that's the mice or the humans.
Full and humans.
Yeah. So it's done in both. That feed-forward response is initially weaker and then gets stronger over time as a training effect with more exercise. What's driving that training effect? Is that still a central component?
That's a great question. I mean, I'm not sure. I think that it's probably an unanswerable question with old experimental techniques. I think that there are certainly now the neurophysiological tools available where in those original laboratory experiments could influence that process and ablate the ability to generate central command. That was the terminology that was used, and see if it becomes untrainable, which I suspect it would be. I think the relationship between that and the fundamental role of parasympathetic function in exercise is rather less clear, but only because we haven't done the experiments. So I think, and actually, then they're not very straightforward experiments to do. I'm not aware of any human interventions where that has been mimicked or provoked, but it's an interesting idea. It's probably a lot of the psychology of exercise is probably very much caught up in that, I suspect. So people who are able to be calm or anticipate the, starting a race, for example, may very well be a key factor. I mean, it kind of makes sense, right? The psychology of it is the idea that you feel good because you want to run hard is quite an interesting one. I mean, it's clearly a fundamental neurophysiological feature. It's not just sort of whimsical idea that I felt like I was going to win. I mean, it's quite a pretty fundamental statement, isn't it?
So I wondered if we could now try and tease out what you've told us about exercise in relation to the concepts that being fit makes you able to tolerate the perioperative period better.
Yeah.
And also the related concept, which is that having an operation is like running a marathon. Are both of those statements true and consistent with your theories?
So I think that the old idea of this stress response, and we're all brought up with it, I think it's a pretty questionable physiological idea, particularly in the modern surgical patient.
I.e. that having a stress response is a good thing or a bad thing?
Or, pretty much all of it, I would say.
Anything.
So I think if you think about the classic physiology we've all been brought up with, that was really based on healthy young people. I'm unaware of any studies that have demonstrated that you have a normal stress response if you have heart failure, diabetes, COPD, and the list goes on. So some work we did as part of the MET study, which looked at functional capacity, including formal measures of exercise capacity. We did a sub-study that really exploited cardiopulmonary exercise testing to ask this question specifically of autonomic function and specific parasympathetic function. So when you exercise, at the end of exercise, your heart rate slows, and the reason your heart rate slows is because you reactivate parasympathetic neurons to slow the heart. And while that's not exactly the same neural substrate as we talked about earlier on in the laboratory experiments, it's a pretty good measure of your parasympathetic fitness. And in fact, we know this sort of by accident because in the early 1990s, there was a couple of really big studies that looked at exercise testing using pretty much the Bruce protocol. They looked at all the variables that generates and found that heart rate recovery at the end of exercise maps to mortality and cardiovascular risk. And what was remarkable about that work, it was published in the New England Journal of Medicine, and what was remarkable about it was that it was repeated in two separate centres pretty shortly afterwards, and they found exactly the same results with exactly the same threshold. Further proof of concept data in humans showed that that was attributable to switching off parasympathetic outflow. So anyway, to cut a long story short, we took advantage of the MET protocol by asking a very specific question, and that was to ask the question, does delayed heart rate recovery map to myocardial injury and complications? Because if your exercise capacity is essentially a biomarker for intact autonomic function, then you would expect an independent marker of autonomic or parasympathetic autonomic function to map to complications. And so this is done in about 1,500 patients as part of the MET study. And it was actually the only independent marker in the entire series of studies that showed a relationship with developing myocardial injury and complications. So very much linked in with the idea that not having an intact autonomic response or specifically parasympathetic response maps to poor surgical outcome. So we've explored that in a number of different ways since, and we've just finished a study where we used ivabradine. Ivabradine is a drug that works on the funny channel. The trial is called Funny, which I'm particularly proud of. The whole concept of this study was not to use ivabradine as a conventional
to slow your heart rate.
It was purely to test the idea that because the channel is only located in the AV node, you can slow the heart, but leave the rest of your autonomic changes during stress and inflammation intact. So it's almost like a human knockout model of the hypothesis. So what we did in that trial, so it was funded by the NIHR as part of my advanced fellowship. And so what we did was, it was a placebo-controlled trial with ivabradine. This is placebo. And we titrated the dose of ivabradine to the heart rate. Basically, it's kind of a weird way to think about intervention in medicine, but it is an experimental medicine study in 350 people with a real-world readout. But the idea behind the drug was to dissect out whether heart rate per se or heart rate as a marker is the important factor, because as I mentioned earlier on, your resting heart rate is determined by vagal tone. So if you slow the heart and it has no effect on your organ injury, then that would strongly suggest the people you're targeting or the people that you're most likely to benefit from or wouldn't benefit from are the ones with poor vagal tone. In other words, you're slowing the heart rate independent of their underlying physiology. Does that make sense? So I haven't got the results for that, sadly, because they're being analysed as we're chatting. But essentially, the work that we did in MET was completely mirrored by some work that was done in Toronto, where they took exactly the same thresholds and found exactly the same relationship. So essentially, this is the proof of concept study with a real world myocardial injury being the primary outcome. Essentially, if you control heart rate in a highly specific manner, so it's titrated to heart rate, it was titrated during the first 3 days before surgery and after surgery. If the hypothesis is right that autonomic dysfunction is a player, then you should see no effect of the drug.
So how does this impact clinicians listening to this at home, or going to give their next anaesthetic in their orthopaedic list next week? What's the implications for their practice from what?
So I think there are two main implications. First of all, I think it does challenge our medical taxonomy somewhat. I mean, if we're saying that the driving mechanism for complications after organ injury is a system you're not even assessing, that has quite profound implications. And it kind of makes sense, right? We know that patients with, if you take the classic RCRI index, no one's ever been able to explain to me why it doesn't matter which combination of factors you get, as long as you get more, there's a relationship with worse outcome. And there's a very plausible and very likely autonomic explanation for that, and that is we know that the commonest cause of autonomic dysfunction is actually hypertension. In fact, there's really good evidence that hypertension is driven primarily by brainstem autonomic dysfunction before anything else appears. But similarly, diabetes and other disease, and deconditioning. If I put you both in bed and put you in bed for 2 days, gave you an isocaloric diet, and then measured your autonomic dysfunction, you would have profound parasympathetic. So it's very easy to lose. I mean, it's not, this is common. As we showed in METs, 40% of the patients in METs had a threshold value of heart rate recovery that mapped in all the previous cardiology studies to death within 3 to 5 years. You can dismiss maybe a 1,500 patient study, but this has been repeatedly shown in the general population. And of course, resting heart rate has got a very strong relationship. So number one, I think we need to think about the medical taxonomy used to assess patients, because it doesn't seem fit for purpose. Number two, we know that complications cluster. You don't go through an operation and just get one organ affected. And so I think, if you work on that premise and you accept that something is driving multiple dysfunction, it's a very plausible idea that what could be a connecting feature of that. And a very obvious place to start is the nervous system, right? And in organs that are commonly innervated. And actually that pattern does bear out. What we found in METs was that exactly that was the relationship. So I guess the immediate impact are there ways we can manipulate this? Well, there possibly are. I think the wider question is, do we understand enough about human organ injury? And specifically, do we understand human organ injury in a very personalized way? So it is highly unlikely that if you have hypertension, very common, comorbidity before major surgery, it's highly unlikely that just because you're hypertensive, you share the same endotype as someone else who's hypertensive. And that has lots of implications for both your propensity to organ dysfunction, the impact of controlling mechanisms like the autonomic nervous system. So I think I'm going to roll you back a little bit and say we need to have a much better understanding of organ injury, particularly in modern surgical patients, because pretty much all our knowledge is based on either volunteer data or young people, and we know very, very little about organ injury in these people. And I think the evidence for that is very obvious in the sense that if you take the same people and look at on a population basis of which of them has poor outcomes from something like pneumonia or UTI or myocardial infarction, they're exactly the same demographic as we see doing badly after operation. So I guess my answer to you would be, I think we need much more mechanistic research into understanding this. I think it doesn't feel or seem right to be doing trials and we don't really understand what the target is. But I think there's a huge scope to take advantage of this. I mean, there are clearly drugs that could be repurposed in a meaningful way. We just have to reconcile ourselves to the fact that we know that neurodegenerative disease is very common. With age, this is a cardinal feature of an aging human, loss of autonomic control. And so perhaps part of this is sort of a big group hug and reconcile ourselves that there are patients in whom these pathologies are very difficult to counter, because there's very good evidence that, for example, vagal motor neurons are quite susceptible to bioenergetic injury. And so, the kind of characteristics of the sort of high-risk surgical patient very much fit with, very impaired. You see this all the time, right? Explain why, you give someone phenylephrine or metaraminol and there's no baroreflex response. That's not just an anesthetic. That is not just an anesthetic phenomenon.
You just keep saying interesting things, and we keep thinking, oh, we want to go this way or that way. But Andy, I think you had a follow-up.
I was going to follow up on 1, 2, maybe 3 points ago now. But you identified this need for mechanistic research or a lack of understanding in that translational gap. And I think actually in many ways that brings us back full circle to how we started this episode. And you said you had 3 hats, one as a clinician, one as an academic researcher, and one as an editor and a publisher. And I'd argue that perhaps all 3 of those groups, not you individually, I think we're both on the same page about this, I agree with you, but I think all 3 of those groups are perhaps missing that important point about the importance of that translational research to a degree.
Speaking with my BJA hat on, I think There is quite a lot of translational work. There's a lot of confusion, especially with clinicians, about what basic science is, what translational work is.
I think we should just take away the basic science term. There's nothing basic about a lot of the work you've just described.
Sure. Well, the exercise bit is pretty basic, but the other stuff was more translational. I think in many ways, that work, there is quite a lot of that work going on. Whether it gets the bandwidth it should is debatable. But I think if you take oncology as an example, personalized medicine is real and happening, and there's no reason why that can't happen in the specialty of perioperative medicine. I think this idea of, well, going back to what I said about, medical taxonomy, it is biologically implausible that hypertensive A is the same as hypertensive B. And we've just we published some work on this with ACE inhibitors and stopping and starting. And part of the rationale for that was based on the pharmacokinetics of the drugs, but also fundamentally based on the fact that not all hypertensives are the same endotype. So in other words, they have biological molecular mechanisms that are fundamentally different, even though we paint them as the same clinical phenomenon. And actually, when we published this work in the BJA, and it was looking at components of the renin-angiotensin axis. And if you do a completely agnostic analysis of the levels of those mediators in the renin-angiotensin system, you see distinct clusters appear with fundamentally different physiology. So, one group in the space study we did had very high aldosterone levels, which interestingly mapped to pretty much what has been described in the general population in the hypertensive endotype groups. And they had 20% higher myocardial injury. So that's kind of what you'd expect from their physiology. So I think the idea of one size fits all just doesn't fly. We know that from other areas of medicine. I think the challenge is to really understand the broad underpinnings of what distinguishes those people. And then, I think there are ways in which that can be approached in a trial design. But I think the three strands all fit together. That work is happening. I think it's often confused with laboratory work. And I think if there's one take-home message from what you just said, is that I think man is the new mouse. I mean, the tools now that are available to do real experimental medicine work in human subjects, even within healthcare systems and healthcare processes, exciting. And I don't think I don't think we're even scratching the surface with biomarkers, for example, to enrich trials, which is a very simple example. And, they're increasingly becoming point of care. So you don't even have to depend on, central laboratories or our lab, William Harvey, to process those samples. So I think there's some pretty low hanging fruit, and I'm pretty excited, I think, about the future of medicine, because I think we're just scratching the surface of that, especially in our clinical area.
Well, you're just so infectiously enthusiastic about this, don't you think, Andy? It's very interesting. I'm going to walk on the Dun-Leary Pier and contemplate my autonomic nervous system, I think, and whether it's functional or not. What about you?
If you can get to the pier, you'll be fine.
Do you think so? Oh, that's good news. Well, we'd really like to thank you for coming on the podcast, Gareth. We'd like to thank the BJA for their generosity in allowing us to interview the interesting people at this meeting. We'd like to thank our sponsors, BD Advanced Patient Monitoring and GE, without whom we couldn't bring TopMedTalk free and open access to the world. We'd like you to subscribe. You can go to our website, our app, X, Twitter, Blue Sky, what else, Andy? Facebook, YouTube, and tell all your friends and subscribe. So we've had a great time here in Dublin, and thanks for listening.
Thank you.
Thanks for downloading TopMedTalk. Don't forget to subscribe, check us out on YouTube, and of course on social media. We're on Instagram, Facebook and X. Also, it's important to remember that TopMedTalk is the broadcasting arm of EPPOM, evidence-based perioperative medicine. We'd love you to find out more about that, and the way to do that is eppom.org. Check out our website and find out about some of the incredible conferences we're going to be arranging across the year. eppom.org.